now that we've seen this pattern, we need some terms to talk about it. It's kind of hard to say a cell that has paired chromosomes versus a cell that doesn't have paired chromosomes and so forth. And so there's also names we want to give to what we call these pairs and things like this. So we have a series of terms here that I'm going to define technically. These are concepts that you've seen before, I imagine. Any cell or organism whose cells are like this, where the chromosomes come in pairs like this, is referred to as diploid, diploid, two sets of chromosomes. So it can refer to cells. Cells can be diploid, but it can also refer to organisms. For example, a cell that's in your body is typically diploid. Not all of them are. But you are therefore considered a diploid organism. Haploid is the concept of having a cell that only has one of each of these types. And you have cells in your body that are like that as well. Eggs and sperm cells in humans, for example, are haploid because they only have one set of chromosomes. So notice what this tells us is something kind of interesting. If you look, there's 23 pairs of chromosomes. Each one is copied. That means this: even though your cells have 46 chromosomes within them, a complete set of human genes is found on only 23. So 23 chromosomes is enough for a complete set of human genes. You have 46 because you have two complete sets. You got two because you have one from each parent. Cells that are diploid have that property. They get one complete set of chromosomes. They have a second complete set of chromosomes identical. Haploid is only one set of chromosomes. So those types of cells are fundamentally different than the diploid cells. And remember, we talked about this chromosome and this chromosome being partners in a sense that they are the same probe is binding to them, which means they have the same basic DNA sequence. These two then have a name. We call those homologous chromosomes. Homologous, the word, comes from the Greek homologous. Homo means the same. Logos refers to proportion or amount. And so what this is basically saying, they have the same amount or that they match. Because they have exactly the same size, shape, location of the centromere, and the genetic content is the same. Even though there are slight differences, again, a few slight differences between these cells' DNA sequences, the vast majority of the DNA is identical. Really, what DNA is a program. It basically tells an organism how to build itself. That program comes to fruition or starts when a sperm cell, which is like, for example, this red thing here, fertilizes an egg cell. At the point of fertilization is when the cells become diploid. This is a haploid cell. That sperm cell is haploid. And this egg is also a haploid cell. So this has 23 chromosomes in a human being. This has 23 chromosomes in a human being. And these things then come together to form a new nucleus. And that new nucleus has 46. It becomes diploid. As soon as that happens, the fertilized egg becomes what's called a zygote. That's the word there, a zygote. The zygote then goes through a series of divisions, which are called cleavage divisions, because it's like somebody takes a cleaver and just chops the cell right directly in half. So for example, right here, this cell has divided into two. It's now what they call a two-cell zygote. Now normally when a cell divides, it's then spends some time growing afterwards. These cells don't. These early divisions, the cells don't grow. They simply divide first, and then they divide again, and then they divide again, and they divide again, and divide again, until they become a solid ball of cells, which we refer to as a morula. It's just a solid ball of these cells. The morula then will start to change. It'll start to go through processes where the cells begin to take on functions. They start to become, for example, cells that will eventually line the gut. They also become cells that will eventually become muscles and connective tissue, and cells that will become the outer portion of the body, the skin, for example, and the nervous system. Once that happens, the morula begins to become a hollow ball of cells. It develops then into this object, which is called a blastula. The morula becomes a blastula because it secretes chloride into its center, and the chloride dilutes the water inside, so the water from the outside goes into the morula through the process of diffusion of water, which you know as osmosis. Osmosis then pushes all of these cells out into a ring around the outside, becomes a hollow sphere of cells. Then the cells begin to differentiate. I'll explain what that means in a moment, but they begin to change their function. Like I said, they start to form the gut, they start to form these other structures. Once they start to do that, then they become what we call a gastrula. The cells no longer are capable of becoming just about anything in the body. They start to specialize into particular functions. Then what you end up with after a time, the gastrula will then start to give rise to structures that you can start to recognize, in this case the placenta and things like this. Once you have that, then you have what we call the embryo. And the embryo, again, sort of looks like this. Here's a series of embryos of different organisms drawn many, many, many years ago, back in the 19th century. These are all organisms of vertebrates. Vertebrates are creatures that have a vertebral column or a backbone. And all of them, notice, are fairly similar. See, they all have a big eye spot here. They all have a tail. You can see the tail segmented right there. They all have these things, these little curly things right along what is going to become the neck. Those are those are structures that are going to become the gills. And then they have a big hollow sphere up here where the brain is going to develop and so forth. They're all exactly the same. So these are all early embryos, and here are late embryos. Now, an embryo is defined as a developing creature that you can't tell the species of unless you're a specialist. So, for example, everything in this row, these are all embryos because you can't really tell what they're going to be. This could be that, could be this. Unless you're a specialist, it's hard to tell what's what. But down here, you notice this has changed, and you can tell that this and this are starting to become more fish-like or more tadpole-like. The transition from embryo to the next stage occurs when you can start to tell what species it is. By definition, when you know what a developing organism species is going to be without being an expert, you can see what it basically is going to become. That's when it becomes what we call a fetus. So, just to finish it off, we have first fertilization occurs, then you have a zygote, which becomes a morula, which becomes a blastula, which becomes a gastrula, which becomes an embryo. I want you to know the definitions and structures of all of those. And then it becomes a fetus when this developing organism is identifiable. So these two you could consider fetuses. These you still can't. They're still embryos. You can't tell exactly what they're going to be unless you're, again, a specialist. Here are the fetuses of all of these organisms. So here, for example, all these are developing chicks. All of these are developing pigs. Here's the developing cow. Here's the developing rabbit. And here's the developing human being. And you can see, interestingly enough, you see this is us. This isn't a human embryo, and you notice it has gills, and it has a tail. Yet, as adults, we don't have gills, and we don't have a tail. Actually, we do. We have a teeny tiny tail. It's called a coccyx. The gill arches actually also still exist. The first gill arch is our upper jaw. The second gill arch is our lower jaw. And a couple of other gill arches are small bones in our ears, and the other ones disappear. These bottom ones disappear in our bodies, and we never develop them. But notice everything here started with gills. But only the fish and eventually the salamander actually use gills functionally as even fetuses or adults. So this is an interesting pattern we're going to come back to. But this is the development of fetuses from the embryos. And then, of course, after we get to this stage here, the fetus stage, then you get to the next stage, which is, for some of us, very important, which is called the tax-deduction. So all of these processes are generated by cells. Cells do all of this. And the question then is, how do you end up going through all of these things from a single cell into a cell that has literally trillions of cells, or into an organism that has trillions of cells in it? How do you do this? There's three main processes by which that occurs. The first, you start with a single cell and you got to make a whole bunch. And again, a whole bunch being trillions if you're making a human being. Person my size, the average size, is approximately 30 trillion cells. So where do you get these 30 trillion cells from? Well, the first process that does that is this process called cell proliferation. Proliferate literally means to make a bunch of new babies. The cell proliferation cycle is something, again, you've probably seen before. It goes through a series of phases. So let's go through those phases. Right here, you have a cell that's just been born, right there where the cursor is. A brand new baby cell enters into a phase that's called G1, or the first gap phase. G you can also think of as a growth, but it literally means gap. In that phase, the cell is basically developing into an adult, a functional cell. But then cells can do something that we can't, and this is something that's really interesting. Cells, as they age and differentiate and become, or not differentiate, but become more adult-like, can then, once they've reached their final adult form, they can stop aging. Not stop growing, but stop aging. That's a very different concept. And when they stop aging, they go into this phase that's called G0, which is a quiescent phase. The word quiescent simply means quiet and not active. That's not true. They're not entirely quiet or active. They're actually physiologically very, very active, but they're not aging. If they're going to age, then they have to get a signal. Some signal from the outside must tell them to start aging. That signal usually comes in the form of some sort of hormone or something we call a cytokine, which we'll talk about later in the course, or some other chemical signal. For example, suppose you have a cell in your skin that's been injured. The cells around it will sense that the cell has been injured and is dying, and then they will get a signal from that cell to start to divide. So what do? The cells that are around that injured cell are in G0. What they then do is they go back into G1 and start aging again. When they start aging again, however, they go to this checkpoint right here. We call the G1 checkpoint. At the G1 checkpoint, the cell starts to age again and double checks to make sure that it's okay. It looks for a number of things. For example, it makes sure that its size is good enough to divide and the nutrients are sufficient to divide. It's got the right social signals, meaning those signals I was telling you about, telling it to start to age again, and that the DNA is in decent shape. If all of that is true, if the cell is in good shape and ready to divide, then it'll go past that and enter into the next phase called S, DNA synthesis. Now, this is critical. Once a cell goes through the G1 checkpoint, it is committing itself to one of two fates. Either it's going to divide and complete the cycle, or it's going to kill itself. There's no other way around it. In fact, that's exactly what the cell does. If it goes out of the non-aging process back into aging and gets to this and discovers its DNA has somehow been damaged, what it then does is commits suicide. Literally, it will kill itself. It engages in a DNA program that will kill itself. Now, if a cell doesn't do that, if it survives that, then it goes into this DNA synthesis phase. It copies the DNA, and I'll show that in just a moment. When it does that, it makes a complete copy of all of the genes. It makes the chromosomes essentially doubled. Then after it gets out of S, it goes into a second gap phase. Now, this second gap phase is a growth phase that's really associated with preparing to divide, because the cell has already committed to divide at this point. So it gets to this point right here. This is another checkpoint. It's called the G2 checkpoint. And what it's going to do at this point is it's going to double check to make sure that all the chromosomes have been completely replicated and that the DNA has not been damaged, and that other factors, for example MPF, which are mitogen factors that basically are signals to itself telling it it's ready to divide, all of those things are present, and it's got everything telling it to do that. If it gets to that point and all of this is good, then it enters into this process called mitosis. If not, then guess what? It kills itself. So this is another dangerous point for a cell. The cell will only go through if, in fact, it's ready to actually divide. It then, if it is ready to divide, it goes into what's called M, the M phase, and mitosis phase. Now, mitosis, you might have heard of, is cell division. Technically, that's not strictly correct. In the profession, we talk about mitosis as really being division of the cell nucleus. That's because a cell nucleus can divide, but the cell doesn't have to divide at the same time. So we separate those two things out. So mitosis technically refers only to division of the nucleus. Cytokinesis is division of the entire cell. The two together, mitosis plus cytokinesis, is cell proliferation. And that occurs here in this M phase. Now, you've seen this before. This is the phase that we like to teach you in high schools and junior highs and so forth, where you start out with prophase and then prometaphase. You may or may not have heard of that one. Then metaphase, then anaphase, then telophase. The metaphase, however, has another checkpoint. Right in the middle of metaphase, and you're going to learn all these phases in the lab, right in the middle of metaphase, the cell goes through a checkpoint. And there it makes sure that all the chromosomes are properly arranged within the cell. They need to all be on what's called the metaphase plate, which is a plane that cuts through the cell and makes sure that all the centromeres of the chromosomes are on that plate. And if it's ready to go, it continues on to anaphase. If it's not, if the cells are not ready, the chromosomes are not in the right position, guess what? The cell kills itself. If all that's good, it goes through, and then the cell divides, goes through cytokinesis, and you end up with two new baby cells, and you go right back into G1. This is called the cell cycle, and I want you to memorize all of that, everything that's happening within that cycle, and I want you to remember what each phase is and what's happening in each phase. And again, every time that there is a place where there's a checkpoint, if the cell does not pass the checkpoint, it commits suicide. That process is called apoptosis, which I'll describe here in just a moment.